Okay, so for the last talk of the day, we have Professor Edgar Delgado Eckert of the University of Basel. So Edgar did his master's in mathematics from the University of Munich. Then he did his PhD at Tufts with a virology group. Then he moved on for his postdoc at, I'm sorry, Edgar, can you just remind?
ETH Zurich.
At ETH Zurich. And currently he is forming his own group at the Children's Hospital at the University of Basel. And his research interests include epidemiology, host pathogen interactions, and some aspects of physiology, which he would mention in his talk. So Edgar.
Thank you very much. So it's the last talk of the day. I hope you had enough coffee or tea so that you stay awake for the next 60 minutes. So what have we done in the last couple of days? There has been a lot of modeling of epidemics of an infectious disease, and we have talked about SIR models, SIS models. And today I want to talk about something different, which I think was mentioned the very first day. I wasn't here yet, which is I want to talk about lifelong persistent or chronic infections. So that corresponds to SI models, but I don't want to talk about these in terms of an epidemic. I rather want to tell you a little bit what we have done using mathematical modeling to understand the mechanisms that certain pathogens use to establish a chronic infection, an infection that your immune system is never capable of clearing. And as I said, I will concentrate on this host pathogen interaction and the mechanisms behind that interaction rather than talking about how such a chronic infection might spread in a population. And so this is a type of pathogens that seem to have one thing in common, which is namely establishing lifelong persistent infections. And one very famous one is Plasmodium, the causer of malaria. And you probably all know that Plasmodium has this cycle of infection, which includes the vector and includes the human host. Just struggling to get the laser to work. Oh, okay. Thank you. So it's this cycle that includes the mosquito, but there is also a cycle within the mammal host, which is this cycle here in blood cells. And there are some other, so I'm mainly interested in what's happening within one host, so that's why we're going to ignore this part of the bigger cycle that includes the vector. And there's another pathogen called Trypanosoma cruzi, which is the causer of the Chagas disease, which is transmitted by the kissing bug. I don't know if this bug is existent in tropical India, but I grew up in tropical South America, and they are all over the place, actually. And so also in the case of this pathology, you have a cycle within the vector, which is the kissing bug, and you also have a cycle within the mammal that allows for this pathology to establish a lifelong persistent infection. Another important example are herpes viruses, which are causes of diverse diseases and are also linked to cancer. So for instance, the Epstein-Barr virus, which I will tell you a lot more about later on in my talk, also manages to infect cells, and those cells go through a process of different migration and differentiation stages, and all those stages appear differently to the immune system, and then the cells finally shed viral particles, which then can infect new cells, so the cycle is closed. So as I said before, what these pathogens have in common are lifelong persistent infections. So if we look at the time course of such an infection within the host, so if we look at the pathogen load within one host as a function of time, so you might be familiar with a curve like this, which corresponds to flu. So you have a peak infection that is then cleared by the immune system. If we look at HIV, you would have a curve like this one, and in the absence of any treatment, then you will have like an explosion in the production of viruses, and the person would eventually die. What I want to talk about today is more pathogens where the curve looks like this. And the question is, what are the mechanisms that allow for such a chronic infection? How can the pathogen actually trick the immune system or evade the immune system so that it's capable of establishing a chronic infection, which also leads to widespread in the population? So if pretty much persons are infected for a long time, they are also capable of spreading the pathogen within the population very effectively. And so this is something that, from an evolutionary point of view, you might think of as a strategy of the pathogens to become very efficient in terms of surviving within a population. And so I was telling you before, something that is characteristic to all these pathogens is this cycle of infectious process within the host. And so we set out to investigate, using mathematical modeling, whether this cycle can be actually the reason or can be a sufficient strategy, a sufficient mechanism to explain chronic infection. And so let me take you through the structure of the model. We have seen lots of models that are based on the same principles, but at the level of epidemiology, at the level of infected individuals being contagious and passing the disease to susceptible ones. So we're going to use the same formalism and the same mass action kinetics and the same assumption of well mixing, but now within the host. So the infective individuals are going to be the pathogens, and the uninfected individuals are going to be cells within the individual. And so the model works as follows. We consider every stage in which the pathogen is. This could be the free virus, or it could be a cell that is infected with the virus and it's transformed into something else. And then we have an interaction with the immune system that tries to control the proliferation of the stage. We have a natural decay of infected cells, and we have an influx from the previous stage. And if we write that using differential equations and assuming well mixing and using this formalism of that is very used in chemistry, for instance, to the mass action kinetics, then the equations look like this. So you have, for the time derivative of the size of the population of a given stage, you have an influx from the previous stage, you have a decay, you have a transformation into the next stage, and you have a reduction that is due to interactions with the immune system. Also, the population of the immune system is stimulated by the presence of the pathogen in the body, so we have to consider the size of the populations of T cells that are controlling the disease, and so this is also based on encounters with infected cells or infective particles. And there is, of course, a natural decay of cells of the immune system. So this is essentially the structure of the model, and let me take you through the specific meaning of the parameters, just so that you keep that in mind for the rest of my talk. So as I said, AI is the decay of a given stage. Fi is the rate at which a given stage is lost to become the next one. Then we have Ri, which is an amplification factor in the process of going from one stage to the next. We have PI, which is the efficacy by which the immune response acts against a given stage. So that is this factor here. CI is the antigenicity of a stage. So that means how visible, how effective is an infected cell or an infective particle in being visible for the immune system. And Sorin B is the natural death rate of the T cell population of the immune response. So this is the essential structure of the model. Something that people usually do to simplify the mathematics of models is that they rescale the variables so that the model is slightly simplified. And so we rescale our variables here, so we introduce new coordinates, and that is essentially working in a different unit. And then we rewrite the equations in terms of the new coordinates. And so you see now the new equations, they have these bars everywhere. And from now on, I will just omit those bars. So something that some of the people who have talked in the previous days, they mentioned is finding equilibria in your model. So this is a very important step when you want to look at, is there a point to which trajectories of the dynamics of your system converge to, and is that point something stable, something attractive, where your system moves towards and it stays there. And so the way of finding equilibria mathematically is that you say, okay, if this is something stationary, then the time derivatives should be equal to 0. So you set your equations equal to 0, and then you solve. And so when you do this, you find one almost trivial equilibrium, which is the uninfected one. So there is no pathogen in the body, and therefore there is no immune response that is specific to the pathogen. And then if you look at less trivial solutions, you end up with what we call infected equilibria. And they can be formally written like that, but you don't need to worry about that complicated formula, because there is indeed only one equilibrium that we will be caring about, which is the one that is biologically meaningful, meaning that the levels of the different populations are actually positive or non-negative. And then we will see that there is also one that is particularly important or interesting, which is a stable equilibrium. But before we do that, we should also talk about R0, which is a magnitude that we have been hearing a lot about in the previous talks. So again, here in this type of model, which is describing the spread of a pathogen within your body, you also have a magnitude that tells you, okay, you got infected, you ate something that contained the virus. Is now that infection going to spread out in your body, or is it going to die out? And you can, within this formalism, you can come up with this magnitude, which will tell you that the infection will spread out if and only if this R0 is bigger than 1. And so what is important, I will tell you in a second what these magnitudes are. Something that we found when we created this model is that you can actually order the different stages of the infection according to what is called in mathematics a partial order. Are you familiar with the notion of a partial order? So partial order is basically the mathematical description of what we do every day when we have a series of objects and we try to order them, for instance, by importance, or for instance by the date. So you can always order them in a way that you know which are the maximal ones, which are the minimal ones. So it turns out that we can do this for the stages in the cycle, and this order is not related to the order given by the direction of flow. It's a different order, so don't be confused by that. So here are the magnitudes that I was telling you about. So R0 is the product of these magnitudes. So they involve the parameters in the model. And then we have sort of a complete product of these guys and then a partial product of these guys, which is between two given stages, J and K. And so by looking at these values, we can establish the partial order. And that's what is written up there. So we say a stage J stars stage K, and we write it like this, if this magnitude is smaller than 1. And so you're probably asking yourself, what am I calling this starving? And the reason is essentially that because the production of a given stage depends on the previous stages, it turns out that you have those big guys, and these big guys will determine how big the population of the smaller guys will be. And this, as it turns out from the analysis, is going to be an important feature of the equilibrium I was telling you about. So when you have this situation, you have that stage J is bigger than stage K according to this condition here. So the regulation of stage J starves stage K of sufficient population to support regulation. So what does that mean? That means that a stage will be called unstarvable if it is maximal with respect to this order. So if we cannot find any stage in the cycle that is bigger than a given stage, then that stage will be called unstarvable. And the other ones for which you can find another stage which is bigger than it, those will be starvable. And so sort of the main result of this analysis was to see that if we look at an equilibrium and it turns out that every unstarvable stage is regulated, then we will call that equilibrium saturated. And so what we found in the mathematical analysis was to say that an equilibrium point is locally asymptotically stable if and only if it is saturated. So it will look like that. If you have an equilibrium, and an equilibrium means sizes, certain sizes of your populations, in that equilibrium in which the stages that are maximal are regulated, so the population of T cells at equilibrium is bigger than 0. And for the ones that are not maximal, that controlling population is actually equal to 0. That turns out to be a stable equilibrium. So what this is telling us, I mean, the mathematics can be a little bit confusing, and you can read more about them in the paper. But going back to the biology of the problem, this is telling us that there is a unique stable accommodation between the immune system and the pathogen. And most importantly, at this accommodation, not all the stages are regulated by the immune system. Rather, it is sufficient to regulate the big guys, according to that partial order that we defined, in order to keep the system in an equilibrium. So what this is telling us is that the steady state configuration of the infectious process comes with a regulation profile, meaning that certain stages will be regulated and some others will be not. This is just a technical detail that I told you a couple of slices before that, we assume the parameters to be non-negative, but now we will allow actually for this parameter to take negative values, which means that there are some stages that are self-establishing. So they also have an internal replication. And this is an important detail because this happens in biology. So you want your model to be capable of describing that situation. And it turns out that then when you include that feature, so this parameter is allowed to be negative, all the previous results remain valid. So now the question is, how useful is this model for describing reality? So I told you we started out with pathogens that go through this cycle. So they infect certain cells. Those cells become other cells, and then eventually they produce more pathogen, and the new pathogen infects new cells. You have this cycle. We have the feature that pathogens that do that tend to be able to establish a chronic infection. We generated a mathematical model that encapsulates all that information, and we saw there is an equilibrium. The model predicts an equilibrium that is stable. So, in which I said there is the trivial equilibrium with no infection, and there is the non-trivial equilibrium where some stages are indeed infected. So this steady state seems to correspond to chronic infection, right? And now the question is, okay, can we actually use this parallelism to describe reality? And the way we're going to do this is we're going to use data from a concrete virus, Epstein-Barr virus, and see how far we can get with this model in terms of making a forecast about certain properties of Epstein-Barr virus infection, which are not pieces of information that we put into the model when we were constructing it. So to this end, let me tell you a little bit more about the Epstein-Barr virus, which was discovered in '64 in Burkitt's lymphoma. So it has been linked to a series of cancers, including 3 types of lymphoma. This virus infects more than 95% of the population. And it's called the kissing virus because people transmit it by kissing. So most people, if they are not infected by their own parents, at the latest by the age when they have a boyfriend or a girlfriend or they get married, at the latest at that point, people become infected. It is usually asymptomatic, but a few people develop symptoms, which is called infectious mononucleosis. You probably know somebody who had it. I mean, it feels like a really strong cold, and some people have to go to the hospital, and it lasts like for about 2 months, and people feel like really, really weak. So as I told you, this is associated with certain cancers. And the main target of Epstein-Barr virus, EBV, are B lymphocytes. There are 6 stages. I will tell you which those stages are. And for each of the parameters in the model that I just described, we have not specific values, but we have a whole interval of plausible values. And that is based on literature or measurements that were done in the lab in which this work was conducted. Also, what is very important, I told you this equilibrium comes with a regulation profile. So you have your steady state, and in that steady state, some stages are regulated by the immune system, some are not. And that is something that, in the case of EBV, is well understood, which are the types of cells or the stages that are normally immune regulated in people that are infected, and which stages are not. So what is interesting about the EBV is that it uses normal B cell development to infect the person. So in a way, it hijacks normal B cells. And this is how it works. So normally, when we talk about how B cells work in your body, so let's say you eat something, and then there is some antigen coming in through your saliva. And that antigen will encounter some naive B cells. Not naive B cells, but B cells that are specific to that antigen, and by encountering sort of the cognate antigen, those B cells will differentiate into what is called a memory cell. And then those memory cells go into the circulation, in which they also proliferate. This is what I was telling you, that this final feature of the model is important. And then you have a terminal differentiation in which those memory cells become plasma cells, and plasma cells are nothing but a factory of antibodies that are specific for this antigen. and they start producing the antibody, which then tries to regulate the antigen. So this is what B cells normally do. Let's see what EBV does. So again, kissing virus comes through the saliva, and then it infects B cells, which then becomes something like a lymphoblast. So it mimics normal B cell biology. And then those cells go into what is called the germinal center reaction. I mean, we don't need to talk about the details of that, but that is essentially a way of selecting more specific B cells to certain antigen. And so it goes through to this stage, which takes place in germinal centers, which are in your tonsils normally. And then you will have within your tonsils, in the germinal centers, you will have a differentiation process which corresponds to the cells becoming like a memory B cell. But this is a process that is now triggered by the virus. And here we have again the same. We have this replication, which also entails that the viral genome is replicated. And then there is this terminal differentiation that is, again, in analogy to the cells becoming plasma cells. And people have identified 3 stages through that. This is called immediate, this is called early and late stage. And then at the final stage, the cell is again like a factory, but not of antibody, but rather of viral particles. And the viral particles, they go out and can go into the saliva to infect new individuals, and they also very quickly infect new cells. And this happens very, very quickly, according to some experiments, so that we don't need to consider the free virus a stage in our model, because either the viral particles are very quickly shed into the saliva, or if they remain in the body, they immediately infect new cells. So essentially, this stage here is not really visible to the immune system because things happen very fast. And there's an interesting experiment that was done in the lab where I did my PhD, where they had people drink alcohol, like 60% alcohol, to rinse their mouth, so to clear all the viruses. And then they took samples of saliva after 10 minutes, 20 minutes. In about 20 minutes, you could see the virus in the saliva again. So this is how fast it is repopulated. So, and then, of course, you have an immune response against all these stages, and that's something that has been experimentally very well characterized, as I was telling you, also in terms of which of these stages are regulated by the immune response and which are not. So in order to validate the model, what we did was the following. I was telling you before that for each of the parameters in the model, we had not a specific value, but rather an interval. And so when you think about each of those intervals, and maybe I can draw it on the board. So if we have here, let's say x, y, and z, and then we say these are 3 different parameters, and I know that the plausible values of those parameters in terms of biological plausibility, it's a certain interval here, right? The same with this one, the same with that one. So this gives us, in 2 dimensions, this square here, and if we look at 3 dimensions, then it would be, you would need to shift this square somewhere to into the board, and you would get this cubus. So this is the cubus in which triples of parameters are contained that are biologically meaningful. And so what we did was, okay, we're going to randomly sample within this cubus parameters and then see what is the model telling us for those specific values that were randomly sampled. So as I was telling you, we have 6 stages. So in principle, there are 64 possible regulation profiles, right? And so what I'm going to do now is I'm going to just code each regulation profile with a binary string. So for example, if I have a binary string of length 6, like this one, a 0 in the ith position means that the ith stage is regulated, and a 1 stands for a non-regulated stage. You could do it the other way around. We just did it like this. And so what I'm going to show you now is we randomly sample a value out of that cubus. We interrogate the model. So the model tells us there is an equilibrium. That equilibrium is stable. And the sizes of the populations, and in particular which populations are regulated by the immune system, that is all a function of those parameter values, right? So we're going to look at, for each randomly sampled family of parameters, which are the regulation profiles that the model is going to tell me. And this is what we do here. So what I'm showing here is, as I said, I code every profile with a binary number of length 6, and that goes from 0 to 63. And now I randomly sample, and this is telling you how often, so this is the relative frequency of a certain regulation profile showing up. Right? So for each point here, that I randomly sample, I interrogate the model. And the model tells me there is an equilibrium, a stable equilibrium. With a specific regulation profile. And so you do this 1,000 times, like we did here, and then you see, okay, the model is telling me there is an equilibrium which has this type of regulation profile. There is one like this, and so you see that there are 4 that are very frequent, and of course you can keep increasing your sample size. And if you do so, some other things that are less frequent will start appearing, but eventually you reach some sort of equilibrium, or you're randomly sampling, so this is something that represents your actual population. So what is interesting about this? I mean, you see there is a clear signal here. I mean, something is going on, right? So I'm representing this again here now with telling you which are the specific stages of EBV infection that turn out to be regulated in those equilibria after interrogating the model. And which is interesting is that 93% of all the profiles that we got from this process of randomly extracting parameters from the cubus are biologically observed profiles. So that is something that when you look at the population of EBV infected people and you measure for what stages are they producing an immune response, there are certain profiles that are common in the population, and this is exactly what we are observing when we interrogate the model like this. There is another way of representing this information. So again, here we have those 4 different regulation profiles that seem to be very common in the EBV-infected population. And you can also look at individual stages and say, okay, out of all those regulation profiles that I obtained from the model, from interrogating the model, how often is a given stage regulated? And so you can generate statistics like this. And something that is interesting is that you see that the memory stage is never regulated, and that is something that is well known from EBV biology. So people have for a long time believed that the reason why EBV was able to establish a chronic infection is because memory is sort of invisible to the immune system. So as I told you, the four most frequent regulation profiles, this memory stage is not regulated. Then we see that memory stage alone is not regulated. That is 54% of the times when we randomly sample the cube. And so there's about half of the cases we just see that memory is not regulated. Then memory plus germinal center stage is not regulated, is about 30%. Memory plus the lytic stages, those stages before the cells start producing more viruses, is about 8%. And then we have this one that is sort of less frequent. You might ask yourself, okay, is this something that has anything to do with these cubos, or could I just go and maybe inspect some other part of parameter space that is here or there, not necessarily in this volume here, and would obtain a similar result? So in order to test for that, we just conducted a permutation test. And the permutation test is that you say, okay, I was talking about parameter A for germinal center stage, and I'm going to say now that is actually the parameter for the lytic stage. So you just shuffle around so that you, in a way, you destroy all that information that relates the parameters to specific stages. And so when you do that, and then you repeat this random sampling, you don't get results like this. I mean, the cases in which you get results like this are sort of rare, and you can calculate an empirical p-value. So it is very rare to obtain a result like this after you have shuffled all your parameters. And one other test that we did was to enlarge these cubos and see what happens. And there also the number of right profiles went down dramatically, which is telling us that so this region of parameter space seems to be very important in terms of tuning the model to describe EBV biology. As I was telling you before, people have thought that it is just this invisible memory stage that allows for persistent infection, and so what we are claiming here is that that is not necessary. I mean, it might be sufficient, but it's certainly not necessary, because you can artificially increase the antigenicity of the memory stage. So you all of a sudden make your memory stage a lot more visible to the immune system by modifying that parameter. And then you can, again, interrogate the model and then see, okay, the model is telling me there will be an equilibrium and there will be a specific regulation profile attached to it. How does that look in that case when I artificially increase memory? And so this is the result. So this is, you get all of a sudden a lot more regulation profiles. You don't get a scenario like this, where four stand out. You get a much more heterogeneous scenario. And it is also a lot more complex in terms of, so this stage is regulated, but it is regulated in all sorts of different combinations of possible regulation profiles. So we get something that it is stable. So meaning that you don't need this invisibility of the memory stage to have a chronic infection. However, the composition of the steady state is completely unbiological. So what we're trying to say here is that we believe, based on this analysis using mathematical modeling, that what is responsible for the stability is not the low antigenicity of the memory stage, but rather the structure of the cyclic infection. We think that the low antigenicity of the memory stage is just responsible for the kind of regulation profiles that you observe. But we think that the stability is more given by the cyclic structure of the infection. So let me tell you a little bit more about validation of the model. So something that we did was to look at the sizes of populations at equilibrium. So the model, when you interrogate the model tells you, okay, you're going to be in an equilibrium in which, for instance, the germinal center population or the memory stage population will be of this size, right? So we went on, and then you said, okay, for all those randomly extracted parameter values, what is the distribution of sizes of the memory stage population that we get? So when we looked at that distribution, that is what is here in green. So this is the predicted size of the memory compartment. We get this green distribution here. And in purple, you see what has been measured in EBV patients. And so what you can see is that these two distributions are, in terms of the center of the distribution, the mean, they are statistically not distinguishable. And I mean, this is something interesting because when we constructed the model, we didn't put in any information about the size of the memory stage in infected patients. We didn't use that information at all. It is actually an information at the level of the population, and our model only concerns, if you will, one person. And however, when we randomly sample out of the cubus of plausible parameters, we in a way obtain information at the population level, which in the case of the memory stage is in very good agreement with actual measurements. And we did a similar thing with the germinal center population. So the size of the germinal center population at equilibrium, at those equilibria that we get when we interrogate the model after randomly drawing a set of parameters. And so what we did in this specific case was to look at those regulation profiles in which the germinal center population was not regulated. So for those cases, which were about 30% out of the sample that we took from the cubus, so we looked at the distribution of population sizes for the germinal center population at equilibrium in those equilibria in which the germinal center stage is not regulated. And we compare that to what is known from EBV positive patients. And so what we see, which is interesting, is that we are at the lower end of the range that is existing in the human population. And this kind of makes sense because when you see that the germinal center population is not regulated, you would expect that that population of cells will be low. Because if you had a very high population of germinal cells, germinal center cells infected, then you would expect the immune response to become active. So what we are seeing here is, we're probably here are seeing those patients in which the germinal center stage is regulated, and the ones here at the lower end are the ones that do not need to regulate that part. So we also found that it was an interesting prediction from the model for the sizes of these populations. So to conclude, so under the conditions that allow for pathogen spread, so R0 is bigger than 1, the model is telling us that there is a stable accommodation between pathogen and immune system. And at this equilibrium, only the maximal stages are regulated. This is something that is also, from an immunological point of view, maybe counterintuitive. So it looks like in an infection that goes through a cycle like this, you can actually get away by regulating the stages that are more important. and the other ones you don't need to care about because those are indirectly regulated through the cycle. That's what I just said. So the maximal stages starve the small ones from having a population that is big enough to trigger an immune response. And therefore, you don't need the direct regulation of those stages. The equilibrium comes with a specific regulation profile. And as you saw, we built the whole story of this work around these regulation profiles. And so we see that the model is capable of reproducing the most common regulation profiles observed in the population. And it makes correct predictions about the size of the memory stage. It makes a correct prediction about the range. And it places the sizes of the germinal center population in the lower end for those profiles in which the germinal center stage is not regulated. And as I said before, this low antigenicity of the memory compartment is not necessary for persistence. Nevertheless, it does determine the regulation profile. And so this is what we think is the strategy, or is what is going on with this kind of pathogens, is that infected stages act as a reservoir for other stages. And that is what makes the whole infection very, very robust. So even if the immune system manages to clear one of the stages, it will be refed through the cycle. So we think that the strategy of persistence is actually the cycle. I would like to acknowledge the people who worked on this project, in particular my former PhD advisor, Michael Shapiro. David Thorley-Lawson is the head of the lab in which many of the experiments or measurements of patient samples were conducted. And Jared Hawkins, which is now at Harvard, he was one of the PhD students at the same time when I was there. I would like to thank the organizers, especially the local organizers, for giving me the opportunity to tell you about my work here. Thank you for your attention.
So how much of parameter estimation do you need to get the match with the experimental data that you were talking about? And it seems like a simple enough model, where basically it goes from stage to stage to stage. So do you need to, have access to experimental data to fit any of the parameters to see that nice match with experimental data that you showed later?
Yes. So the difference between some other approaches that people perform is that we did not classical parameter fitting. What we did was to take from the literature intervals of plausible values. So we didn't say the parameter should have this specific value, or I'm going to tune my model so that it reproduces some data. We didn't go down that road. We said we want to have knowledge about the parameters that is based on completely independent experiments, and then come up with a cube of plausible values, and then we're going to interrogate the model based on randomly sampling that cube. So you see the difference. We didn't do any fitting. It's sort of a different type of approach.
But within that cube, you're uniformly sampling, or do you sample more from the center, less from the periphery? I mean, do you do any kind of bias sampling?
No, we did a completely blind sampling out of the cube. I mean, we thought about doing something more sophisticated based on some previous knowledge, but we didn't get very far with that. So we decided, okay, this is going to be a really completely blind sampling of the cube.
More questions?
So the other thing I wanted to tell you, I was asked to tell you a little bit more about the research in my group at the University of Basel, and also because we're currently offering a job. So just very quickly, what we do in my group, it's not only related to host pathogen interactions, but we are also doing a lot of physiological work and work in cohorts. So for instance, we have projects in which we assess the effect of tobacco smoke and air pollution on cardiac activity. We have a project in which we assess the temperature control in children and babies that come to the world too early. We also look into therapy response in asthmatic children. We have a big project which is about phenotyping in complex diseases such as asthma. So maybe you know a little bit about asthma, but asthma is a very heterogeneous disease, and not every patient responds equally to treatment. So you try to subdivide your patients into different categories, and that's what they call phenotyping. And so we have a computational approach to phenotyping. That is one of our big projects at the moment. There is also another project that I started during my postdoc, but I've been wanting to keep working on that, and I will tell you a lot more about that during the conference in a couple of days. So, and that is discovering biochemical signaling networks by studying gene interactions and epistasis. So essentially, the fact that you have different genes that influence a process, and you have some sort of robustness there, in the sense that if you mutate the gene or you actually completely remove it, the process remains working. And so that's something that people call gene interactions, or they call it epistasis. And so we have developed a formalism by which you can look at epistasis data and then come up with biochemical networks that may not be known to biologists. And finally, what I talked about today, we're also interested in mathematical modeling and computer simulation of host pathogen interactions. And so we are offering a postdoc job, so please, if you are interested, and if you have sort of a computational background, but also some solid work in biology or physiology, please visit our website and apply. Thank you.
Thanks. Just one question about the earlier slide. So the disease phenotyping, this is machine learning type of classification?
Yes, that's using pattern recognition algorithms, machine learning algorithms, yes.
So what is this complex data over which you do the classification?
So what we tend to do, because phenotyping classically has been done mainly on the clinical side, where there are a lot of a little bit subjective criteria. So the doctor looks at you and then he thinks, I think you are this kind of patient. So it's not very reproducible. So our idea is to use really objective information. And so what we have been using is, we have access to some cohorts where people, children or adults, they took a device home in which they blow. And this device measures your lung function. So it measures, for instance, your peak expiratory flow. So when you're blowing out, what is the highest flow of air that you can produce with your lungs? And so we have time series of such values over 4 to 6 weeks. And so we analyze those time series, and using machine learning algorithms, we try to group time series that are similar.
So let's thank Edgar again for a nice talk. Thank you. Now, and see you at dinner.